cellular respiration. My goal in this video is to help you not only get to a point where you can describe what happens during cellular respiration, but to get you to a point where you truly understand why each step of cellular respiration occurs. In order for you to understand the detailed steps of cellular respiration, you first have to have a solid understanding of the big picture. Let's first look at why cellular respiration is important. First off, all organisms, including bacteria, protists, which includes organisms like paramecium, amoebas, and algae, plants, fungi, and animals like you and me rely on cellular respiration for energy. Protists, plants, fungi, and animals all have structures in their cells called mitochondria where cellular respiration occurs. So we will only focus on how cellular respiration occurs in organisms that have these mitochondria, and we will not talk about bacteria. What is cellular respiration? Cellular respiration is the process that organisms use to convert the chemical energy stored in sugar molecules like glucose to a usable form of chemical energy called ATP. This process requires oxygen and is therefore also called aerobic respiration. ATP stands for adenosine triphosphate. It is called a triphosphate because an ATP molecule contains a chain of 3 phosphate molecules. Now you may be wondering, well, why do cells need both glucose and ATP? Why can't they just use glucose directly to do things? Well, that's a good question. Did you know that one molecule of glucose contains about 100 times more potential energy than one molecule of ATP? It would seem like glucose would be a better direct source of energy for cells to do work. But the truth is that glucose contains too much energy for cells to use directly. If cells try to break down glucose rapidly to do cellular work, most of the energy would be lost as heat. It would be very difficult for cells in our body to capture and use that energy before it was lost. ATP, on the other hand, contains less potential energy per molecule and is a much more unstable molecule than glucose. So it is much easier for our cells to break down ATP by breaking the chemical bond holding the third phosphate group on. Our cells can then use this energy to power chemical reactions and movement within our cells. Here is a great analogy to understand this concept. Imagine that glucose is like a $20 bill and ATP is like a quarter. If you go into a coin laundromat with a $20 bill, you would not be able to use it to wash your clothes. You would first have to convert that $20 bill into quarters in order to do anything with that money. Although each quarter has less value than a $20 bill, they are perfect for turning the washers and dryers on. Just like quarters, each ATP molecule has just the right amount of energy to do work in your cells. Whereas glucose, like a $20 bill, is much better for storing energy, transporting it, and delivering it into your cells. Now that you understand why ATP is important, we have to discuss two terms that are necessary when discussing both cellular respiration and photosynthesis. These terms are oxidation and reduction. It is important that you know what these words mean because it is likely that your textbooks will throw these words at you and expect you to know their meaning. I experienced this when I was a student. If you don't understand these terms, then you will become lost really quickly. So what do these terms mean? Well, the reactants in a chemical reaction can either be oxidized or reduced. We say a molecule is being oxidized when it loses electrons. How can you tell if a molecule has lost electrons? Usually, a molecule loses electrons by losing hydrogen atoms. This makes sense because if you remember some basic chemistry, hydrogen atoms are simply made up of a single proton and a single electron. If, for example, a molecule like methane lost its hydrogen atoms, then we would say methane is being oxidized. On the other hand, we say a molecule is being reduced when it gains electrons, usually by gaining hydrogen atoms. This might sound confusing that a reduction means gaining electrons and oxidation means losing electrons. But an easy way to remember this is memorizing the acronym OIL rig. Oxidation is loss, reduction is gain. Let's look at the equation for cellular respiration to further understand this concept. Let's write the equation for cellular respiration. Get a piece of paper and write this equation down with me. Remember, first you are writing down the inputs, or in other words, the reactants of cellular respiration. These are the molecules required for cellular respiration to occur. If this is hard for you to remember, then just think about how you feed your cells. What do you eat and what do you breathe in? You consume the sugar glucose in your food, and you breathe in oxygen gas. These are the reactants in cellular respiration. The reason you need to breathe and eat food is because you need to provide the cells in your body with the resources they need for cellular respiration. Next, what does cellular respiration produce? In order to help you remember this, just think about what you breathe out, because you breathe out the waste products of cellular respiration. You breathe out both carbon dioxide and water vapor. You can witness this if you exhale on a cold day or exhale onto a glass window or mirror. And don't forget that this process also makes ATP, which your body uses for energy. To be exact, each glucose molecule can be used to produce up to 38 ATP molecules. If we were to balance the equation for cellular respiration, then we would just add a 6 before the oxygen, carbon dioxide, and water. Now I want you to try to answer a question. Where do the carbon atoms in the carbon dioxide that you breathe out come from? If you look over here at the reactants, we started with glucose and oxygen. The carbon atoms could not have come from oxygen gas you breathe in because oxygen gas does not contain carbon atoms. So they had to have come from glucose. This means that as glucose is broken down during cellular respiration, the 6 carbon atoms that were in the glucose molecule are released as carbon dioxide molecules. This means that you breathe out the carbon atoms in your food. I think that's pretty crazy to think about. This is why when you lose weight, most of the mass is actually lost in the air that you breathe out. So if the carbon atoms in glucose are breathed out as carbon dioxide, then what happened to the hydrogen atoms that were in glucose? They had to have gone somewhere. The only product of cellular respiration that contains hydrogen atoms is water. This means that the 12 hydrogen atoms that make up the 6 water molecules produced during cellular respiration had to have come from glucose. What actually happens is that these hydrogens were removed from glucose, and then they were added to the oxygen atoms from the oxygen gas we breathe in. Therefore, this means that the oxygen gas that we breathe in turns into water in our bodies. Hopefully, this has already cleared up a common misconception. Many people think that the oxygen gas we breathe in is turned into carbon dioxide gas, when in reality, the food we eat turns into carbon dioxide gas, which we exhale, and the oxygen gas we breathe in turns into water in our bodies. Now that I've blown your mind, let's get back to oxidations and reductions. Since glucose lost hydrogen atoms to become carbon dioxide, has glucose been oxidized or reduced? Remember, oxidation is loss, reduction is gain. Since glucose has lost hydrogen atoms, it has been oxidized, turning it into 6 carbon dioxide molecules. What about oxygen? Since oxygen gas gains hydrogen atoms to become water, was oxygen oxidized or reduced? It was reduced. This is why we call cellular respiration and photosynthesis redox reactions, because one of the reactants is reduced and the other is oxidized. Okay, now I think we have built a strong enough foundation to dive into the steps of cellular respiration. Cellular respiration has 4 main stages. The first stage is called glycolysis. The second stage is the preparatory reactions. The third stage is called the citric acid cycle, but you may also see it called the Krebs cycle or the tricarboxylic acid cycle or TCA cycle. but I will refer to it as the citric acid cycle for the rest of this video. And then the final stage of cellular respiration is called the electron transport chain, often abbreviated as the ETC. The goal of the first 3 stages of cellular respiration is to completely break the glucose molecule down, whereas the goal of the final stage of cellular respiration, the electron transport chain, is to produce a majority of the ATP that is made during cellular respiration. As I said, the goal of the first 3 stages of cellular respiration is to break glucose down. But remember how I said that our cells cannot break down glucose rapidly because most of the energy would be lost as heat? So our cells must break down glucose slowly. During cellular respiration, our cells will break all the carbon-carbon bonds in glucose, and special molecules will carry away the hydrogen atoms. In order to carry away the hydrogen atoms during cellular respiration, our cells have special molecules called electron carriers. The electron carriers that are used during cellular respiration are called NAD+ and FAD. NAD+ and FAD differ in the number of electrons and hydrogen ions they can carry. NAD+ can pick up 2 electrons and a hydrogen ion, and when this happens, it becomes NADH. Notice NAD+ has been reduced. NADH will eventually travel to the electron transport chain where it will release its hydrogen ions and electrons. If NADH releases its electrons and hydrogen ions, then it has become oxidized and it will become NAD+ again. Side note, remember that hydrogen atoms consist of a single proton and a single electron. If I remove the electron from a hydrogen atom, then I'm left with a single proton, which we call a hydrogen ion because it has a positive charge, and I'm left with a free electron. Now let's get back to the electron carriers. The second electron carrier is called FAD. FAD can pick up 2 electrons and 2 hydrogen ions, and then it becomes FADH2. FADH2 will also eventually transport all of its electrons and hydrogen atoms to the electron transport chain. When it gives up its electrons and hydrogen ions, it becomes FAD again. Now, let's draw a diagram of the steps of cellular respiration together. If you are watching this video to help you prepare for a test, then you will want to grab a piece of paper and draw the next diagram with me. Let's first draw the cell membrane. All of cellular respiration is going to happen within the cell. Although cells can have anywhere between 100 to 1,000 mitochondria, we are going to draw one large mitochondrion so that we can see the processes occurring inside of it. The mitochondrion actually has 2 membranes. This is the outer membrane. The inner membrane has a lot of folds on it to increase the surface area for ATP production. We are also going to draw the electron transport chain now because the molecules involved in the electron transport chain are found in the inner membrane of the mitochondria. The electron transport chain consists of a set of molecules. As you will see later on, electrons will be donated to these molecules from NADH and FADH2. The molecules in the electron transport chain will pass the donated electrons from one molecule to the next down the chain. The energy from the movement of these electrons is going to have a crucial role in ATP production. One of the most important molecules involved in the electron transport chain is called ATP synthase. ATP synthase is an enzyme that will produce a majority of the ATP during cellular respiration. Although there are multiple electron transport chains and ATP synthase molecules along the inner membrane, we will only draw one set. Cellular respiration is going to begin in the cytoplasm. The first stage of cellular respiration is called glycolysis. If you break down the word glycolysis, glyco means sugar and lysis means to break. So glycolysis means to break sugar. During glycolysis, glucose, which is a 6-carbon molecule, is broken down to 2 3-carbon molecules. each called pyruvate. Notice that although we split glucose, we still have a total of 6 carbon atoms, and no carbon atoms have been lost yet. Glycolysis actually needs a little bit of energy to jumpstart the entire process. 2 ATP molecules are used to initiate glycolysis. But because glycolysis also produces 4 ATP molecules, we get a net gain of 2 ATP during this stage. The production of ATP during glycolysis is called substrate-level phosphorylation. This process uses special enzymes to create ATP by adding a phosphate to an ADP molecule, which stands for adenosine diphosphate. Another thing that occurs during glycolysis is that 2 NAD+ molecules grab some electrons and hydrogen ions. This reduction creates 2 NADH molecules. You will notice that all NADH molecules that are made during cellular respiration will head to the electron transport chain because they will be used to help mitochondria make lots of ATP. After the 2 pyruvate molecules are made in the cytoplasm, they are going to travel inside the mitochondria. The space within the mitochondria is called the matrix. This now begins the second stage of cellular respiration, called the preparatory reactions. The preparatory reactions are going to prepare pyruvate for the next stage of cellular respiration. During the preparatory reactions, each pyruvate molecule is broken down further. Notice that we are slowly breaking down the carbons that originated in the glucose molecule. During the preparatory reactions, one of the carbon atoms from each pyruvate is removed. This creates a 2-carbon molecule called an acetyl group. This acetyl molecule attaches to a molecule called coenzyme A, which is why we call each of the 2 molecules produced during the preparatory reactions acetyl-CoA. So what happened to the 2 carbon atoms that were removed from the pyruvates? They are actually released as carbon dioxide molecules. Mitochondria produce 2 carbon dioxide molecules during the preparatory reactions, 1 molecule for each carbon atom removed from pyruvate. One last thing happens during the preparatory reactions. 2 NAD+ come in and 2 NADH molecules are made. Where are these molecules going to go? Just like the other NADH molecules, they are going to head to the electron transport chain where they will be used. Now, the acetyl-CoA molecules are going to enter the next stage of cellular respiration. I'm only going to draw one acetyl-CoA molecule. However, both of these acetyl-CoA molecules will go through this cycle. One of the first things that happens during the citric acid cycle is the coenzyme A will leave. Then the acetyl group will attach to a carrier molecule, which is not drawn in this diagram. When the acetyl group attaches to the carrier molecule, a citric acid molecule is briefly made. During this process, the carrier molecule itself is recycled and reused, which is why we call this stage the citric acid cycle. For each glucose molecule that enters cellular respiration, the citric acid cycle occurs twice, once for each acetyl-CoA molecule. The acetyl group is then completely broken down. Therefore, the last carbon-carbon bonds from the original glucose molecule have been broken. Since each acetyl group has 2 carbons, 2 carbon dioxide molecules are produced for each acetyl-CoA molecule that entered the citric acid cycle, for a total of 4 carbon dioxide molecules produced. It is important to note that the citric acid cycle produces the most electron carriers of any stage of cellular respiration. For each acetyl-CoA molecule that enters the citric acid cycle, 3 NADH molecules are produced. These 3 NADH molecules are going to make their way to the electron transport chain. The citric acid cycle also produces a little bit of ATP using the same process we saw in glycolysis, substrate-level phosphorylation. This is just a fancy way to say that a phosphate is added to ADP using an enzyme. 1 ATP molecule is produced for each acetyl-CoA molecule. As you can see, the citric acid cycle makes the greatest amount of carbon dioxide molecules, the greatest amount of NADH molecules, and is the only stage where FADH2 is made. For every acetyl-CoA molecule, one FADH2 molecule is produced. And FAD comes in and carries away some electrons and hydrogen ions to become FADH2. FADH2, like NADH, will go to the electron transport chain. Now let's look at how many NADH molecules were created altogether. We created a total of 6 NADH molecules in the citric acid cycle, 2 in the preparatory reactions, and 2 in glycolysis, for a total of 10 NADH molecules. We created a total of 2 FADH2 molecules during the citric acid cycle. Now let's focus on what happens in the electron transport chain. The first thing that NADH and FADH2 will do is give up their hydrogen ions and electrons. Therefore, they become oxidized back to NAD+ and FAD. The hydrogen ions are released into the matrix, and the electrons are going to be donated to electron carriers in the electron transport chain. The electrons that are donated to the electron transport chain get passed from one molecule in the chain to the next. The movement of the electrons creates enough energy to actively pump hydrogen ions from the matrix into the space between the inner and outer membrane. This creates a large buildup of hydrogen ions in the intermembrane space. When there is a large concentration of ions on one side of a membrane, this is called an electrochemical gradient. An electrochemical gradient stores a lot of potential energy. The energy stored in this gradient is going to be used to make significant amounts of ATP. Before we talk about how all that ATP is made, let's step back and look at the electrons that were moving through the electron transport chain. When these electrons get to the end of the chain, they need something to pick them up and carry them away. The last electron acceptor in the electron transport chain is actually oxygen. What this means is that the oxygen that you breathe in eventually makes its way into the mitochondrion so that it can pick up electrons from the electron transport chain. At this time, it also picks up 2 hydrogen ions, therefore turning oxygen into water. Oxygen is crucial in driving the electron transport chain because without oxygen to carry the electrons away, new electrons would not be able to enter and move through the chain. Oxygen is great at receiving electrons because it is very electronegative, which means that it has a strong attraction for electrons. Remember, the movement of the electrons was used to create the large hydrogen ion gradient, which is also called a proton gradient, visible in our diagram? The mitochondrion is going to use this gradient to make ATP. The way it will do this is by opening a channel in the ATP synthase molecule. If this channel opens, where will the hydrogens want to flow? They will flow back into the matrix. The movement of the hydrogen ions back into the matrix causes ATP synthase to actually rotate, like a mini turbine at a hydroelectric plant. The rotation of ATP synthase causes phosphates to be added to ADP, therefore creating about 34 ATP molecules for every glucose molecule that was broken down. When ATP is made using an electrochemical gradient, this is called chemiosmosis. This is different than the way ATP was made during glycolysis and the citric acid cycle because chemiosmosis uses the energy from the electrochemical gradient to power the production of ATP. Now I hope that you have a better understanding of how everything that happens during cellular respiration leads up to the production of ATP.